Thank you, Chairman, for inviting me, and thank you to all of you coming to listen to us this morning. As Stefan said in his quite thorough and interesting talk around some of the molecular and translational scientific developments, the context matters. So what I will try and do, for those of you who are not deeply embedded in oncology as it is practiced, is to set the context of what is happening in oncology so that we can then see where innovation may be needed. The bad news is that cancer is getting more common. In our lifetime, half the people in this room will develop cancer. Now, that's not meant to scare you, it's meant to be a reality that we face. And about half of that increase in incidence, just over, depending on which cancer, is due to the fact that we are better at treating other diseases, so we live long enough to get a cancer. But that's also a challenge because it means that the age of the patients we treat is also increasing. The workload in our cancer hospitals continues to rise. I can't comment about surgery, I'm not a surgeon. But I know in many hospitals, in the day chemotherapy units, the annual increase in patient attendances is between 5 and 10%. We don't have a 5 or 10% increase in staff every single year. And the reasons for that is that we are better at treating patients with advanced cancers. Fantastic. We have new drugs, new approaches, the patients live longer and get more treatment. We are also treating more patients with early cancers, with drugs as well as surgery and radiotherapy. And, of course, the whole volume continues to increase. The second reality is workforce. Clinical staff, whether they're pharmacists, nurses, radiographers, doctors, we are not cheap to train. It's several years of undergraduate, postgraduate training. You cannot turn on a workforce in 1 or 2 years. We need to plan 5, 10, 15 years ahead. And in many countries, workforce planning is not very good. And increasingly, and I'll give a couple of pointers to this later, patients want and are right to want to be treated by an expert workforce. They expect us to know more and more about the diseases we're treating, and that takes time. And of course, our economies are not behaving the way we want them to. So we have a difficult challenge. More work to do. Most of it is good stuff for patients, not necessarily enough people to do it, and not necessarily enough money to do it either. So let me paint 2 very quick scenarios, and they're meant to be somewhat of a caricature. 20, 30 years ago, as an oncologist or a surgeon or whatever, I might treat many different kinds of cancers. I would make my decisions on my own. I might occasionally ring a friend. And I would follow up my own patients. I would have this little empire of my patients, and I'm the best person to know what to do with them. And it probably wasn't very difficult, because with 1 or 2 exceptions, there were very limited number of treatments. It's not difficult to know how to use 6 drugs. It's more challenging to know how to use 60. And now, rightly, we've moved to multidisciplinary teams where you may have 5 or 10 different experts making a decision in one disease. Instead of one person making a decision in 10 diseases, you have 10 people making a decision in one disease. So, for example, in my own institution, we see 700 new breast cancer patients a year. We've worked out that we have the equivalent of 1 whole-time senior clinician in MDTs every week of the year. That's a resource that is well placed, but it's expensive. And of course, we have many new treatments with many newer toxicities, cardiovascular, immunological, and of course the risk for interactions. So the system is getting more complicated because of innovation. Stefan, this is a coincidence. I hadn't seen Stefan's slides. Stefan looked at something, the revolution at a molecular level in lung cancer. When I started in oncology, the the question that was asked about lung cancer was, is it non-small cell or small cell? And that was fundamentally your treatment distinction. There was modest benefit from chemotherapy. A small proportion of small cell patients with limited disease could be cured. Most radiotherapy was palliative, and a small number of patients could have curative surgery. It wasn't easy to treat lung cancer, but the spectrum of what you had on offer was fairly limited. Now it's completely different. You need a multidisciplinary team. You need to understand the EGFR mutation status of your tumor. You have a series of drugs in that space. We now need to know about the ALK status, and we have crizotinib as first line. There are second line drugs coming in. We have a range of chemotherapies, just getting approval for the anti-PD-1 drugs. Even lung cancer has got a lot more complicated, and yet we still have yet to make dramatic differences to outcomes there. But there are innovations away from the molecules that need to be made. So, coordinated approaches to care. If I see a patient, do I get every single piece of information about that patient, even in my own country's healthcare system? No. I get much of it. I don't get all of it. There's still a lack of accurate and precise information sharing. Multidisciplinary working. Is it absolutely standard for every cancer patient across Europe? No. Why not? Before we're worrying about next generation sequencing of every tumor, what about multidisciplinary assessment of every patient? Quality approaches to diagnosis. If I get a pathology report from another hospital, do I know that that's correct? Well, depends which hospital. Depends what quality assurance process is in place. Is that standard across Europe for every molecular marker? No. And there's a strapline which has come through in the UK, and I can't translate it to all the languages, but patient involvement is key. We used to be very paternalistic. I think you should do this. The strapline that's come through is, from a patient perspective, no decision about me without me. You don't make the decision, we make the decision. Is that universal in Europe? I don't know, but probably not. So we already have innovative approaches to cancer care that we have not completely implemented. So that's at the system or systematic level. Obviously, at the individual patient level, we have a number of important developments. Breast cancer is the disease I know most about, so I tend to talk about it because my knowledge of other areas is shrinking. So we measure hormone receptors in breast cancer. I can tell you in the UK, that was a battle to become standardized because one or two key opinion leaders said it was irrelevant. Now, I think pretty well standard across most of the world where it's possible. HER2 testing is pretty well standard in breast cancer. It's coming through in gastric cancer. And there is a message of context. Because HER2-positive gastric cancer doesn't have the same implications as HER2-positive metastatic breast cancer. You may use the same class of drugs. They may be effective. But the natural history of the diseases is not the same. So the context, even for one molecular test, matters. RAS status for the use of EGFR inhibition in colorectal cancers is becoming standard. It's the wild type that matters, not the mutation. And, of course, for inherited genes, BRCA1 and BRCA2 testing, important for a patient to know their risk of developing cancer. It's also becoming important for knowing how to treat the cancer. How universal is that across Europe? And importantly, are the guidelines for getting the tests the same? Nope, not even in my own country. So I can see a patient in Edinburgh who we will get a BRCA1 test on. In another city in Edinburgh, the geneticists say, nope, her risk isn't high enough. The risk is irrelevant. She has breast cancer. This is a test about how to treat her. So even in one country, we don't have a universal approach to making available stuff that is now relatively straightforward. And of course, we're moving into an era of multi-parameter tests. Again, I quote the breast cancer ones. These are prognostic, primarily. They may have some predictive relevance for use of chemo. At least 3 out there, Oncotype, Mammoprint, PAM50. They will come through in other diseases. Are they universally used in the same way? No. Now, Stefan gave a very good molecular basis of personalized or precision medicine, but you need to know what you're looking at. So, sorry about this. These individuals all have the same gender. They are all wearing the same headgear, both in terms of shape, color, and probably manufacturer. They're all carrying the same thing in their right arm, an umbrella, essential in the UK. And they're all wearing a dark business suit. You need to know where to look. The difference is, of course, whether they've got stripes. Take a different set of people, and you need to look at a different characteristic in order to personalize them. You can't just say, this is the approach that works. So for some tumors, mutation sequencing matters. For some, it's copy number variation. For some, it's protein expression. And I'm sure Stefan is right that for others, the microenvironment, the tumor niche will matter. We don't yet know which one needs which approach. And if we look at the wrong thing, they all look the same. And we miss that opportunity for a personalized approach. But personalized or precision medicine needs to be achievable. If it involves 16 different biopsies of a patient with metastatic cancer, is that really deliverable in every hospital in Europe, let alone the world? It's not going to be. There are patients for whom biopsies are more dangerous, either because of their comorbidity or because of the location of the tumor. It's much easier to take a biopsy from the surface than deep in a viscera. And we need to know how to use the data. So a precision approach has to be achievable, not necessarily in every patient, but across the board, to be implementable. It has to be affordable. And we cannot escape the fact that the costs of delivering healthcare are rising faster than the resources made available to deliver healthcare. So whole genome sequencing used to cost millions of dollars for one sequence. We can now do the wet lab sequencing for around about €1,000 plus. So in theory, it's probably the cost of doing a PET scan. So in theory, we could sequence every single cancer in Europe. What we don't have are the trained bioinformaticians and clinical geneticists to interpret the data. And they, I'm afraid, will cost an awful lot more than just a few more €1,000. So we can do some of the technological developments, but have we got the system in place and the trained people to interpret the data? And Stefan's hinted at this, and I think this is also important. The cost of drugs is rising faster than inflation. Are they worth that extra cost is an important question. And I think personalized care has to be acceptable. It's not just for the young, who are fit and can deal with the additional tests, and it should not just be for the rich. And when we deliver a personalized approach, the efficacy, there's a strange word in there for those who are not English speakers, don't use the word outweigh, it's not one I recognize. It's my typing. Wasn't a deliberate joke. The efficacy has to justify the toxicity. Just 2 more weeks in progression-free survival for toxicity may get your drug to the market, but actually are you giving what patients need? Now, this is an example of why the regulators, to my mind, need to be part of the conversation. So again, HER2-positive breast cancer, metastatic, and previously treated with Herceptin. GSK had a drug, now called lapatinib, which looked like it worked in this situation. So they go to the regulators, the FDA, because they're based in the US, and say, we want to demonstrate efficacy. And they said, well, the only approved drug in this setting is capecitabine monotherapy. So combine your drug with capecitabine, show it's better, and we'll give you a license. And that's what they did. And I was one of the 2 CIs on the trial, so I'm conflicted here. Left-hand side, add lapatinib to capecitabine, and you get better control of the disease. On the right-hand side is a trial run in Germany, same control arm, capecitabine, but what they did was what we were doing in the clinic, which was adding Herceptin to capecitabine, because we had some phase 2 data to suggest it works. The axes are different, and I tried to fiddle around getting them to look the same, but it distorted the graphs. Add Herceptin to capecitabine, and it also works. So which one do you think we use in the clinic much more? The right-hand one, because it's less toxic. But the regulators were not interested in doing a comparison against the real world, because it wasn't in the regulators' world, because it's not an approved therapy. And if you look across cancer, you will find other examples of where a regulator's approach to the right trial to prove efficacy is not actually reflecting the question I want to know in the clinic and my patient wants to know. They would like to know which of the two superior treatments is better. I know the answer in terms of toxicity. I don't know the answer in terms of efficacy. This also caused a headache for health technology appraisers because, of course, the health technology appraisers could only compare against capecitabine monotherapy because that's what's in the trials. So the regulators here were imposing an approach which worked as a scientific experiment. Yes, it's better, but actually didn't address the real question in the clinic. So reimbursement is a challenge. When, I notice I put the word when would it be appropriate to spend 10,000 euros a month on a drug? The answer is maybe sometimes it is. But when I started in oncology, people were complaining about drugs that were costing 100 euros a month. So you can see the price inflation has gone way up for lots of reasons. I'm not challenging the reasons, I'm asking the question is, when is it right to spend that much on a drug? What benefits do that drug actually bring? The clinical trials are a carefully designed, usually very well conducted experiment. Compare A with B in that setting, in those patients, with those precise criteria, and you get an estimate of the benefit and an estimate of the toxicity. Take that to the real world, and it may behave very differently. Sometimes it turns out to be a lot more toxic. Sometimes it turns out to be a lot more efficacious. Herceptin, arguably, in metastatic disease, is probably better than the trials estimated. Fortunately, we have access to it. And if a patient comes to me and I say, you should have this treatment, how sure am I that he or she will get those benefits? Because, in fact, that patient would have some particular aspect which would have meant they were ineligible for the trial, even had it been running. So do I know that that result translates to this broader population when I don't know their comorbidities, their concomitant medication, and I certainly don't know their genetics? So what other innovations can we do? And Stefan's hinted at this, and I think there is a big opportunity across Europe. I suspect most hospitals prescribe their chemotherapy electronically. All will prescribe their radiotherapy electronically. In many parts of the world, you're prescribing in the community from your general practitioner. Even if the prescription isn't electronic, the pharmacist will have an electronic record. We'll have electronic hospital records, and we can, but rather intermittently, we can collect patient-reported outcomes electronically. They can do it on a smartphone, they can do it over the web, they can do it sitting in clinic. We could capture an enormous amount of data about what we're doing today electronically. And yet, most of the time, we don't use that. We may do specific audits, but we don't look across the whole picture. And why do I think it matters? Well, actually, how do we treat patients now? Are we using our current therapies in the optimal way? Probably not. How well do those treatments actually work? Do they deliver the benefits they're supposed to? Patients ask us, and unless we've done an audit, we don't know the answer. And if we're coming in with an innovation, in the trial it was compared with drug Y, which is the approved medicine in the US. Actually, the Europeans may completely ignore that or may not always have access to it. So in fact, what would that drug replace? What is happening now in that population of patients? And would we use it in the same sequence? Does the sequence of drugs matter? Probably, but not always. So, last couple of minutes. Where next? The first innovation, I think, is to do what we can already do better. There's an awful lot about what we know works in cancer care that is not standard across Europe. Multidisciplinary working, specialization, etc. There's also stuff we all do, which in fact is probably a waste of money, time and resources. So, breast cancer again. There's very little evidence that seeing your doctor once you've been treated for breast cancer has any difference on your outcome, and probably very little difference on your quality of life. And yet, in different ways, in different places, we do different amounts of follow-up. Why? Because some people think it's a good idea. Actually, it's probably a waste of resource. Research is good. That's fine. There's data that if a hospital is research active, all the patients in that area benefit. Not just the ones in the trial, 3–5%, but the whole cadre with that disease. It's true for ovarian cancer, colorectal cancer, cardiovascular disease. So why do we not embed research in our hospital healthcare systems, and indeed our primary care health systems in Europe? Because that would improve outcomes. Research is seen by some as a special, extra sort of luxury. It shouldn't be a luxury. It should be there for everybody. So measure what we're doing now, and we might be surprised. Now, this came out coincidentally in The Lancet very recently. I haven't put the whole editorial up, but I think the top heading is really important. This is looking at a global perspective. Access to medicines. The Lancet is arguing the status quo is no longer an option. There needs to be a change in a system so that people worldwide can get access to medicines. I think there are other innovations that we could already do that we're not putting in place. We need innovation to implement previous innovations, whether they're medicines, multidisciplinary working, whatever it happens to be. And finally, who are these stakeholders who need to be involved in this conversation? In the drug approval world, it probably feels to many clinicians that these are the two parties that matter. And yes, they're important. The regulators need to approve, and those who develop drugs need to develop them. But actually, health technology appraisals is becoming critical. If you have a fantastic drug that costs £1 million a day, not many people will get it. And actually, you need to build into your trial designs, I would argue, or your post-trial designs, a measure of just how good that innovation is. Very often, the model by which a drug comes in through the regulatory process doesn't fit with what people do in the clinics. So I think we need a bigger circle here to involve those who do the treating. And yes, there's a gap in the middle, because sometimes it feels that's what's happening. That's for whom all this is about. We need to put them back in the center and think, what is it they need to know? What's the information that they should be made aware of that can be gained from many sources to implement innovation? Thank you.
Thank you for the invitation. So what I decided to present to you is actually some of the learnings that we have done over the past few years. And that's why I have a little bit this long title, which is Optimizing Patient Outcome by Leveraging Learning through Big Data and Clinical Research Platform in a dual longitudinal continuity framework. And I would like to introduce you to this concept of dual longitudinality. I promised Stéphane that I would not speak about the EORTC, and I would not even speak about our precision initiative platform, which is SPECTA. But what I want to tell you is that the concept I'm going to show you come out of this volume of expertise. EORTC has a huge volume of clinical research across Europe, and in the last 3 years we have, I would say, piloted the possibility and try to explore solution how you run a multidisciplinary, a multi-tumor precision oncology platform. What I'm going to show you, again, this concept of dual longitudinality is not only outside of my head, outside of the head of Denis Lacombe, but we have challenged this concept with partners. The partners have been, of course, on the other side of the pond, because if we would have to find solutions and change profoundly the way we do things, it would not only have to be one country, a few countries, but it would have to be global. So the concept I'm going to present to you has been a challenge with our partners like the USNCI. We have been discussing also with people that you know probably at the MIT and the new DIGS, the new drug paradigms, with Cancer Commons. Also, actually, we are preparing back-to-back publication on their ideas and our ideas. But also, there has been a consultation process in Europe, and I have exchanged this idea with IMI already, with EMA, and actually, this presentation was made also last week in the context of the PERMED from the DG Research to all the European representatives of the member states. So the concept has been challenged, and I think I will a little bit accelerate on the rationale, because a lot has been said. I think you are all aware about our challenges, and something that came really evident for us in all these, especially last year, expertise of running, again, international platform, is really that we don't have mechanism at this point in time to take probably what is one of the major challenge for cancer, which is this tumor heterogeneity and escape mechanism. And this has been said in the previous presentations, because actually we work in silo and patients go from one silo to the other without any bridge. And what I'm going to say, what I will try to demonstrate to you this morning, is how we need profound transformation actually to have these links between the silos. I think there are a number of recurrent pivotal questions that we are addressing in this transformation. Is that, of course, the classical phase 1 to 3 model, is it still adequate? How do we access? I think innovation is not an issue, but access to innovation is an issue. There are a number of challenges, actually, which were addressed a couple of weeks ago in a regulatory science meeting that we have addressed with the regulator, with USNCI and AACR, in terms of quality assurance requirement, in terms of all the issues for pre-analytical and analytical. How do we develop this biomarker? What is the regulatory acceptability of the target, and so on? So how do we take that collegially in a world that is constantly evolving? Because what we have to prepare also is the fact that our regulatory environment is going to change in the coming years. We are heading up to a new, to 3 regulations, which are the data protection regulation, the IVD regulation, and the clinical trial regulation. So we have also to anticipate how we are going to run these programs in this rapidly changing environment. So basically, what we have been trying to understand and to gather as expertise is a little bit to address this challenge. How do we completely revamp the clinical research so that we move towards a data-driven healthcare, basically from omics to economics, again, embracing all these challenges that you see here, with the central point is the demonstrating clinical utility of biomarker. But as you can see in the bottom of this this chart for you industry, how do you address business risk reduction and improve quality assurance? So this is really what the parameters of the transformation, the new type of clinical research that we see, and your challenges as industry to bring new drugs to the market. So I thought this was also very interesting, a little bit to bring some rationale to the presentation, is that these are recent data from earlier this year from the Biotechnology Innovation Organization, where we see actually that still in oncology we have a higher attrition rate than in other disciplines, which is part of the equation that we have to take into account. But the next part of the equation, which is a good problem to have, is that as compared to other disciplines, in oncology, there is a strong pipeline. So it's a good problem to have. You see actually here the number of transition of molecules that transition from the various stage of drug development. But indeed, how do we address this amount of volume that comes to us very rapidly? How are we going to organize this clinical research? And we see that when we speak to you, to industry, the competition that there is around, for instance, the new molecule. So I think there is a need to rationalize and a little bit to structure things. And also, I got this slide. They are very Dutch-oriented because I got this slide from Jaap Verwey. But basically what it shows you, and again it's been said by the previous speaker, it is basically the cost per quali and the acceptability, if we look at the old cytotoxic drug, by the NICE in yellow, by the US government, by the Dutch government, and of course completely outlier, what is the acceptability of the US medical oncologist. And of course, if you look already at not so innovative, but class of agent, the monoclonal antibodies, you see evidently that the acceptability by the payers is much lower. So I think all these parameters are there. How are we going to address, and have we substantially changed the way we do drug development and clinical research to address all that? So a number of years, 3 years ago, we have already published that, which basically was our vision about changing the clinical research pathways. And actually, as you can see, this Diabolo shape represents a little bit the new challenge and the vision, in the sense that we have to early on to understand the biology of disease and the mechanism of action of new drugs. We know already that the drug approval process and the pivotal trials are changing dramatically, and we have trials that are smaller but much more complex, technology-based. But of course, we see also that there are new challenges in real life, and how these treatments are actually embedded in the standard treatment. And based on this, we have developed new programs. So SPECTA, the precision medicine, is one of those. But we are also piloting what is the role of the other stakeholder in Europe. And for instance, on the real life issues, we are already developing collaboration, for instance, with the registries to understand actually the external validity of clinical trials. So what can we do to address this gap? So what I want to tell you is that at this point in time, we have developed a new type of expertise where the various stakeholders come around the table, around clinical research, to address these challenges. Is it structured? No, not at this time. It's individual discussions with different stakeholders, but again, trying to structure things around a changing clinical research pathway. What we wanted to do, actually, is to have new continuity solution that span from proof of concept into effectiveness. And that's one of the dimensions I'm going to show you, but this dimension is purely drug-oriented. It's a drug development. It does not take into account the dimension of the patient. So basically, this heterogeneity and the fact that we don't have the mechanism to properly address actually recurrence and resistance. And I believe that what we are missing in Europe, what we don't have, is addressing this second dimension. So again, selected but non-exhaustive challenges. I think what I want to highlight is mainly the first line, is that we continue to write protocols the same way we've been doing. We write a protocol, and then we go out to search for patients that meet the eligibility criteria. And we see the difficulty in a hyper-fragmented population, the difficulty that you and us have to find these patients. So the idea and the center of the transformation would be, can we think about another way of doing things and reinverting, having patients who have the full information about their disease that would be seeking protocols, and would that appropriately impact on the access and on the feasibility of clinical trial. And basically, what I want to tell you is that I think the mechanism that we use are the archaic ones, and we have not thought enough about the new mechanisms to circumvent that. So we have a number of issues that you have also identified, or they've been already alluded by the previous speakers, is that we have a suboptimal anticipation of the real-life questions. We don't know optimally how to address the number of combinations that will come in real life. The number of combination novel that you have in your silo remains extremely small, when sometimes there is a very good rationale to move very early on. And we have inappropriate setups for long-term outcome research, while actually the patient needs this continued evolution, because they have a disease, they are going to recur once, twice, eventually they will go in long-term follow-up, and they need this solution and they need this approach so that they have a guarantee for taking the completely, I would say, the complete longitudinal aspect of their disease. So big questions that we've been addressing is how do we reconcile the continuum of care of the patient and the continuity of solution for drug development. And this brings me a little bit now to talk to you about this principle of dual longitudinal continuity. And what you see here actually is you recognize the Diabolo, so this new vision, but which is only one dimension, which is the one of the drug development process. And you have these patients that enter into one specific drug, one specific development of one drug. But the issue for this green patient is that he's going to recur, and he's going to go into another protocol or into, I would say, a standard treatment. And we have no way today to understand the mechanism of relapse, because this patient go from silo to silo. So basically what we do not have is solutions that are going to address that. And you can see, actually, you can multiply that. Of course, what is important is to guarantee to any patient to have this capacity of understanding the longitudinality, and they can benefit from different treatment, whether they are routine, whether they are in advanced development or in early development. So I think that's something that we are missing if we want to really implement an agenda of precision medicine. I would like to give you an example, for instance. What we see today is basically companies that develop different drugs in silo, so very parallel. And we have a number of discussions. I go to many meetings where we speak about the payers, have to speak to the regulators, to the drug developer, and so on. This remains, again, extremely drug-centered. I never hear, wherever I go, solutions that are patient-centered and that will allow the patients to have the information that she needs to actually anticipate the potential evolution of the disease. And the researchers, they come to us, our investigator, they say, we have real-life issues. How we are going to combine this drug? So I think one of the reasons also why we need a profound transformation is that a number of these questions have to be anticipated. And to be anticipated very early on, I think we need, again, other mechanism, because these challenges are there, and when these real-life issues come to us, it's quite often very late. And we are left with many questions and many years of research. So the transformation proposal that I would like to discuss with you is how are we going to build longitudinal large-scale data capture platforms that are constantly curated and annotated? And we need extremely large data, and that's why this concept has been confronted with the North American solution, and that's what we wanted to pilot with our precision medicine Specta, is the feasibility of such platform in an international setting. So basically, you can imagine that if you have the capacity, again, taking into account that you need high quality clinical, biological imaging data with treatment details and outcome, of course, which is not a lot of data, based on very, I would say, solid quality assurance program, then you can already see how you can address the challenges that I've depicted early on in the slide that I've shown you a few minutes ago. I think that if we want to move to this transformation and have this approach, there are two specific assets that need to be developed. I will not speak to you this morning about all the operational and the regulatory innovation that we need in Europe. We've been through how you pilot, how you conduct such a platform in Europe. There are a number of regulatory issues. I decided not to speak about it because it's a presentation by itself, but there are challenges to that. What I would like to discuss with you and show you is probably more the new clinical research architecture and what would be the capacity and what would be potential the delivery. I think that if you have such a platform, and we already have this type of, I would say, output that are being piloted, is that then you can much more easily do multiple new drug development because you are under the same platform. You can apply at any time of the drug development. You've seen this little patient in the Diabolos that can come at any time of the drug development. I think what is important and what we see also is the standardization of methodology, design, endpoint, population, technology. We see a vast heterogeneity in the approaches, which makes extremely complex then I think the full field and how to make a decision. What I think is what we could do, what we could think if you have this central point to, I would say, give to the patient this full identity of their tumor, is that then you can have shared control population, and also to imagine then that you have contemporary benchmarking type of population. You have a permanent capacity for enrolling in clinical trial, and also the capacity to do sometimes complex trial like basket concept, adaptive design, and multi-arm, multi-stage trials that are very difficult to do in Europe. They are feasible in some of the countries, like in the United Kingdom, but it's extremely complex to do in Europe because of the fragmentation and because also the absence of such solution at the international level. And of course, then you would have, of course, a lot of capacity for efficient data exchange, again, because you would have a centralization of the data. So basically, that would be the way it would function. If you have an incoming flow of patients where you can have this high quality data, you put patient at the center. Then it gives to the researchers the opportunity to understand the biology, to all the time re-question when new knowledge comes in, and this is already happening actually in our environment with Specta, that you can speak with technology developers because of course you have access to biological material, outcome data, and so on. A subset of these patients are real life. They come in real life, and you can imagine that a subset will be amenable to clinical trials, and then this subset, of course, is very interesting for drug developers and for regulators, and how you can therefore optimize access to clinical research. But we still have a number of questions that come to us. Is we cannot randomize, there are multiple combinations, and so on. So how are we going to benchmark in situation where we can bring patients to clinical trial, but maybe we cannot randomize. We are more in the real-life type of setting. But if you have this incoming flow of patients with this data, then I think what you can see is that you can use also with new methodologies these incoming patients, those that are not in clinical trial, that are contemporary with same type of quality, you can use them as benchmarking and discuss that also with the HTA bodies and the payers, how you can provide some benchmarking evidence. And this discussion have started also, because I think here we can bring an added value. And actually, if you take this chart and if you look vertically at the 3 columns, and you see under this diabolo that comes back, you see that with the first arm, actually you address knowledge development and early drug development. You can come with some type of conclusion with the subset of patients you bring into clinical trial, and then you can have some benchmarking for effective implementation into real life. This can work, but of course there are a number of challenges ahead of us to address, and that's what we've been piloting over the years. So I think what could be the expected deliveries is an alignment of competency of the stakeholder, a rapid identification of patients or group in a pre-competitive manner, and I think that's where the IMI instruments are very interesting to expedite the start of clinical trial, connect more efficiently the knowledge development to real life, because again, you empower this diabolo shape to happen while you put patient in the center. I think a more an easier capacity to benchmark technology, to benchmark population, to standardize the methodology, and of course to follow the patients on the long run, so to put more effort on long-term outcome research across data set. And for instance, like immunotherapy, I think personally I have a question. There are many immunotherapy trials today. Who is going to actually to do the long-term toxicity, the long-term pharmacovigilance and assess the safety profile in years from now after the drugs are on the market? So I believe that the ultimate need is independent data capture for all type of clinical, biological, and imaging data and records alongside biomarker test results and all therapies that patients have received in databases which are constantly curated and annotated. Again, that's the principle that we have developed. That's the principle that we are benchmarking with other parts of the world, and that we are at the time maturing. I think that we have reached a point where we have identified the gaps, but of course we need now to transform this knowledge. We need to transform this, we need to make this possible at a larger scale, and that's where I believe a partnership, and you've seen this slide, that all the stakeholders can benefit from such platform, but it requires a collegial willingness to completely change the way we are doing things. Sorry, I'm going the wrong way. So I'm not going to describe SPECTA, but basically if you have questions, that's the way it functions. Today, SPECTA is completely integrated. It's not even a program. We are integrated completely into the ORTC procedure, and that's where I would like to finish. Indeed, so it functions as a prospective and longitudinal clinical research annotated biobank where we can ensure this patient dimension, so this second longitudinal dimension of the chart I have shown you earlier on. And I would like just to finish to a little bit advertise our molecular targets and cancer therapeutic meetings that will occur in a couple of months from now, where, of course, big knowledge is going to be communicated. With this, I would like to thank you. I hope I was clear enough. Thank you for your attention.
Denis, very nice talk. Mark Lawler, Queen's University, Belfast. One question in cancer is the lack of a cancer ontology. So actually our way we describe cancer so that we can capture from electronic health records, etc. We're just not very good at that. And I just want to know what your thoughts on that, and maybe we can learn from, for example, rare diseases where they are very good at that.
Yeah. Yes, that's a point we are addressing with all how to implement the health technology, the electronic health records, and how we are going to move to new type of description of the disease. That's one of the challenges we need to address. I can tell you that the way we have selected to perform SPECTA at this time is not optimal. That's not the one we want, but that's the only one feasible in Europe. We are exploring other routes, actually, for instance, partnership with registries, which have all the patients, and how we can start actually accessing to the patient, not necessarily for a clinical research platform, but from population platforms. And here I think we would gain a lot of, I would say, leverage to address this type of questions.
Maybe also looking at something like HPO or
Yeah.
NCI storage as well.
Yeah.
Thank you. I will address a different gap in the current infrastructures in Europe that have led to our initiative to create Cancer Core Europe. So Cancer Core Europe is an initiative to pave the way for a multi-site virtual European Cancer Institute. It takes us from new treatments and principle of concept studies to outcome research. And the key word is sustainability, because when we look at the history of initiatives in Europe, in the FP6 program, there was a program called Eurocam Plus, and it was basically an inventory of barriers, duplication, fragmentation of cancer research in Europe, and to see how we could come up with solutions. So we had many fora to discuss. We went across all activities in cancer research in Europe, and indeed found, of course, that there's a tremendous amount of duplication. Some amount of duplication is not a bad idea, because you may find out that you need it to prove that something actually does not work or does work. But there's a lot of fragmentation. And the recommendation that came out of that Eurocam Plus FP6 program was there's a big lack of mutualized translational research platforms in Europe. Create a TR platform and see how you can move forward with that. So identify the link and create a link between basic and clinical research. So that led to the creation of Eurocan Translational Research Platform, which was an FP7 program. Now there you see there were 26 partners, and there were 16 work packages. And you may already get a sense of the common denominator in a lot of FP programs that may not always lead to sustainability, because the principle often has been that we sort of assemble around an idea and around an opportunity to get money to launch that idea, but that you will not necessarily find 26 partners who know each other very well and who will be very successful to really work together and then stay together. And so a common denominating observation is that a lot of networks or FP programs have led to 4 years of working together, more or less, but that when the money runs out, a lot of things evaporate after that. And in the end, that's not how we progress and how we advance. So the recommendation out of this Eurocan Translation Research Platform was create a core. Create a core that first needs to establish itself as a core and then slowly expand from there, because it's just too complicated to get everybody together around multiple working packages, etc. And so we created Cancer Core Europe. And look at the first word. It's auto-financed. So these are 6 institutes, and I'll tell you which ones, that put their own money and invested in creating a virtual 6 institute, 1 institute philosophy. And that in and by itself is a big change, because you only do that with the vision to create sustainability. Otherwise, you will never do this. So create the core and expand later, but make sure that you have the core first. So I don't think you can immediately work with everybody on every topic. I think you need to pick your partners very carefully to first create a core program. And so among the best, here are 6 of the best in Europe. And there's a reason why these 6 chose each other. So we have Cancer Center Cambridge, Deutsche Krebsforschungszentrum and the National Center for Tumor in Heidelberg, Gustav Roussy, Karolinska Cancer Institute, the NKI Amsterdam, and Vall d'Hebron Institute of Oncology in Barcelona. And in a way, it started between Gustav Roussy and Heidelberg. And many stories start also because of some people knowing each other very well and knowing each other's institutions very well and actually envy each other's institutions for very peculiar reasons. So now, if anybody who has ever been to DKFZ and NCT in Heidelberg knows that the phenomenal infrastructure and the critical size in basic research there is of course, the big beacon. And there is no lack of money, so to speak. So it represents a force that you are not going to find in a freestanding cancer center, which is Gustave Roussy. But when you look at Gustave Roussy, we have a phenomenal critical mass in patients. We have the biggest clinical research program and early clinical trial program in Europe, and we created the infrastructures to have mutualized translational research platforms, because within that size of an institute, you can avoid that departments take their own routes and sort of split off, and that people identify with their apartment. No, everybody identifies with the institute, and therefore you create all these mutualized platforms. And so where you have one institute where 30% of patients go into research, into clinical research, and you have the other institute which has this phenomenal basic research infrastructure for imaging, for radiation therapy, for sequencing, for, you know, whatever, you name it. You know how you envy one another. And that's sort of how it started, and we said, okay, we need to create a core, but pick our partners very carefully, because we're willing to put up our own money and share data and share our culture. So what you see then is that you have three moguls and you have three freestanding cancer, comprehensive cancer centers. So Val d'Hebron, Gustave Roussy, and KI, Amsterdam, are three freestanding cancer centers. What is the common denominator there? Very well developed clinical research, very well developed translational research, and three of the 5 largest early clinical trial programs in Europe. That's one force. You have Cambridge, you have Heidelberg, and you have Karolinska. Well, I do not need to explain what that represents. So what it represents in total then is the research continuum that you need nowadays to interact with biotech, with high tech, with pharma, to say we can identify programs where we can create the continuum, not only around the early clinical phase trial portfolio and have the critical mass of patients to feed that system, but where we have the translational research platforms inside our institutes to accommodate all these studies, which are now biomedical experiments. It's got nothing to do anymore about old-fashioned toxicity assessment of yet another chemotherapeutic agent, and where the questions actually, based on patient biopsies of whatever type, is feeding into a system of basic research platforms that need, in their totality, need to cover the whole field. And so when we talk about proteomics, well, everybody knows Matthias Uhlen in the Life Science Institute in Karolinska. It's like the prime site, perhaps in the world, for proteomics. And so proteomics at Karolinska and Heidelberg is phenomenally strong. Is proteomics strong at Gustave Roussy? No, because we have other platforms in the basic research area, which is mainly immunogenic cell death platforms, apoptosis, autophagy, and basic tumor immunology platforms. That is our strength. But we cannot also have proteomics. And we have actually hired in firm into our buildings to do all the sequencing, to roll out the clinical and personalized cancer medicine program, because we could never build the 40 plus last generation Illumina platform that Heidelberg has, because that kind of a platform we, as a freestanding cancer center, we cannot put up. So as long as you see the complementarity of the philosophy, you have 3 moguls and you have 3 large freestanding comprehensive cancer centers. So this was launched in now exactly 2 years ago. But it goes here. So it needs to be a transformative initiative to create the sustainable, integrated research care capabilities and continuum. And with the vision to create a multi-site European Cancer Institute. So what do? We carry out translational clinical research, conduct next-generation clinical trials in the personalized cancer medicine, molecular medicine domain, and in immunotherapy, but also in radiation therapy in combo with immunotherapy, etc. Develop, therefore, personalized cancer medicine, establish standardized academic diagnostic platforms, obviously, because you need to have the same readout in the various institutes, create large shared databases. And here shared is the big thing, because how are you going to share? You're only going to really share if you know each other well and you actually have signed not just a consortium agreement, but now the creation of a legal entity. So this takes 2 years to work this all out between the 6 institutes, but now we are a legal entity that will share our databases, not just at the electronic dossier or patient file level, but also at the other level behind, because 30%, 25% of our patients are in research programs, and thus there's yet another level of data that needs to be shared. How are you going to start and create this kind of a thing with 26 centers? Forget it. It will never, ever happen. But if you have 6 and it's a legal entity and all the rules are clear, then the next member can live up to those rules and apply. So that's the philosophy. And in the end, when you create this with the critical mass that we have, you also will create a platform for outcomes research. And I'll come back to that point. So I've talked already about the complementarity, right, of the 3 and the 3, basic element. I've said something about critical mass, but just take a little look closer at the critical mass. We see 60,000 newly diagnosed patients per year. We treat up to 300,000 patients per year, and we see in consultation 1.2 million patients per year. Now, with a common IT system, and for that, we need, therefore, to share our clinical databases and research databases behind it, and, of course, share our research projects. You create prospectively defined, fully clinically annotated databases, which you actually need in a longitudinal fashion to do outcomes research. So there's a lot of talk about outcomes research in oncology, and it stinks. And it stinks for the reason that it's like Swiss cheese. Not that Swiss cheese smells bad, but Swiss cheese has a lot of holes in it and very little cheese. And this is our problem in oncology. And that's why we stink at doing outcomes research, because the data are not there. Why? Because it involves always 3 or 4 or 5 disciplines. Pathology is often not there or poorly described, a lot of question marks. The radiation, the imaging is full of holes. Then you have surgeons, you have radiation therapists, and you have a medical oncologist. And that's only already in the discussion of the primary tumor. And then the thing relapses, the whole thing starts all over again, and you think that is all documented in the databases? Come on, it's not. It's just full of holes. It's a totally disastrous situation. So you can say, well, we still are going to do it because big data will solve it all. This is baloney, because big data of a lot of holes is just big data on one big hole. And so we will learn nothing by the simplification of thinking that big data is going to solve it. Why does big data work in hypertension control? Because it's so simple. You have blood pressure, you have 3 drugs, and you have the computer of a general practitioner. So outcomes research in hypertension control, yeah, that I believe in. But outcome research in oncology needs something fundamentally more savvy in the way it's created and set up, and therefore it will only be successful in prospectively defined, fully clinically annotated databases. And behind that, actually, we will have also research layer database behind it. So one legal entity on this slide, that's the word. And why is that? Well, because in the beginning, it's sort of difficult to even talk about this. But then in the end, when we talk to all the biotechs and the pharmas and the high techs, etc., the recurring question was, wait a minute, whoa. We want one door to knock on. So it was like when Kissinger said, when you call Europe, who picks up the phone, right? So this is the same question. So there's one phone, there's one portal, and there is one legal entity. And this is not what we do by creating consortia. And that's why the stability and the sustainability is not guaranteed through consortia. Of course, we need to have our common SOPs and platforms, and there we are developing common validated for the genes molecular diagnostic platform and common immunomonitoring platform, because we have some exceptional talents in those fields. Well, when you look at the molecular diagnostic platform, all you need to look at is what Matthias Uhlen, what Heidelberg and Cambridge already can define there. And when you look at immunomonitoring platform, when you look at Guido Kremer and Laurent Sietvogel and Gustav Ricci, when you look at Ton Schumacher in Amsterdam, and Dirk Jäger in Heidelberg, you will have your common immunomonitoring platform. So from early clinical trials to outcomes research, I talked about it. So we have one portal, we have one legal entity, and we have 6 working groups. I listed it here on one slide. The most important working group is how to resolve and create the platform for data sharing. And so there, what's of great help is that Gustav Ricci and DKFZ NCT Heidelberg, they already started with a big project with SAP. And SAP is the computer system that rules the financial world. And the reason is it's their system that has the biggest operational online memory capability that allows you to open up 40 screens on your screen and look at silos at the same time. And this is actually what we need because of the prior talks, the complexity of fragmentation, molecular splitting up of tumors, and all the data silos that we have in pathology, in molecular diagnostics, in imaging, et cetera, behind each patient. And so the doctor should not have to go to a statistician to say, well, I have this question, but the doctor should have direct access. Well, I'm talking now about a select group of researchers, MD PhDs, who are trained in this field, and they can open up 5, 6, 7 silos of data on patients at the same time, ask questions, and you get your graphs and you get your readouts, and you can actually ask the next question, rather than to having to go to the statisticians who say, well, it's an interesting question, come back in 6 weeks. I may have an answer. This is not how we can proceed anymore, and we cannot be staved off anymore by old-fashioned Ayatollah statistician attitudes like, oh, wait a minute, what you're doing, because you have not upfront already corrected for multiple comparisons and all that, I think it's illegal. No. Our young researchers, between 25 and 35, that's where they should have free access and exploration to databases that are, of course, fully anonymized, and explore and explore and explore, because they all think faster, better, and more originally than most of us in this room. It's like that. So empower the young people in your institute. So data sharing, common diagnostic platforms, functional imaging. And then we take and we offer to biotech and pharma, et cetera, we offer actually from preclinical development, because we have very big preclinical development facilities, into early clinical trial development, because that's where a lot of questions are, both in molecular medicine as well as in immunotherapeutics. And then I talked about the databases. And then, of course, there's always a program where you need to get the young people rotate, get together, organize summer courses, summer schools, and things like that. And that's the 6th point, training. which means I'm going to skip all these slides, and you'll be happy about that, because I dealt with the basic elements. I just want you to understand what's the initiative. So it's auto-financed, but we already have a number of successful grant applications with EIT Health, Kick In Alive for personalized cancer medicine biomarkers, TransCan project on heterogeneity in breast cancer and ovarian cancer. The Agence Nationale de Recherche in France gave us a grant for mobilizing young PhDs, labs, etc. And of course, we will prepare a big immunotherapy project for IMI. We have particular strength there. What have we already launched in terms of clinical trials or are just about to launch? So we're doing the Poseidon trial, which investigates PI3 kinase inhibitors in luminal breast cancer. A basket of baskets trial, which is a multi-layered basket, and which is a thing that only works if you have a large panel of phase 1 studies to accommodate new targets and new drugs. So at Gustave Roussy, we have 75 simultaneously ongoing phase 1 studies. Val de Bron is somewhere up there as well. Amsterdam has about 60. So we, and Heidelberg now is at 30, so we have 150 different early clinical trials to accommodate all these new targets and targeted therapies. And we have particular strength in immunotherapy, actually where we launched the SABRE PDL-1 trial, so it's stereotaxic radiation therapy in metastatic cancers in combo with an anti-PDL-1. Obviously, in the philosophy that when you treat, you can use actually the immunogenic cell death capability of radiation therapy as a priming of the immune system, which will then be unblocked at the peripheral level by anti-PDL, etc. So it's clear that these kind of protocols are launched by us and are then co-financed and financed by industry. Same thing is for the development for intratumoral protocols, because there is a presentation system that's actually intact inside the tumor. For liver mets, oxaliplatin is the immunogenic cell death inducer, and then it's to be followed by intralesional checkpoint inhibitors and the potentiating effect of toll-like receptor inhibitors. So, in the end then, because we're talking about Europe, it's about inclusiveness. But, as I said, you first need to create the core before you can become further inclusive. Otherwise, you're headed for failures on the sustainability chapter of this initiative. And so now we have our legal entity, now everything, the rules are set, the working groups have outlaid their programs, etc., and now we're able to very, cautiously expand. And obviously, Italy is the number 1 on the list with the Istituto Tumori in Milan to join us. But it's a high bar that we have created. So there's a number of things that need to be regulated. So this is it. The objective in the end is to be a precursor of a virtual European Cancer Institute. We will never have a physical NCI in Europe unless we build it in Liechtenstein under tax-free conditions. But even there, I'm not sure that will happen. And so it's a virtual thing, and we believe in it. Thank you very much.
How do you fix the data sharing across country borders in your initiative? If you can collect data across, there is the data privacy laws differ.
Sure, yeah. But the way we, by fully anonymized systems, can share data that already does exist. So they are not traced back. They are only traced back to patients within each institute if necessary, but not on the shared platform that you can't have. But you don't need it to do the research that we need to conduct.